your IPA. So you can get out of this window and open up your IPA.
Your screen's in the way.
Oh, dang it. All right. At least I didn't spill anything on my computer this time. Does everybody's list, so you should open up your project manager. You should see this ABC GCB. It should be in bold. Double click on that.
Is anybody still cycling? All right.
See, I did that first this time. Last time, yesterday, like people's gene lists were still not getting done. So what this is doing is, bioinformatics is, at its heart is, what are the odds that I get something contained, this certain amount of genes that we have collected, that we know share something? What are the odds that I pull all of this stuff just, in, say, any gene list, right? The reason this works is I have a limited things to choose from, right? If you could think, say, picture all 30,000, human genes as green balls. And say I have a pathway or something, and it's, there's 40 members in that, 40 pink balls. And in our gene list, we have 748. So I get 748 draws out of that 30,000 balls. What are the odds that I get 1 pink ball, 2 pink balls, 3 pink balls? The more pink balls I get, the more that's not an accident. And we can define these relationships as any way we want. Okay, so let me open this up. This first window is just basically kind of your greatest hits of your gene list. When I teach this to high school kids, this is how I frame it, is that basically, activated B-cell lymphomas are crime. We did some investigation. We got a bunch of people that were around the crime when it occurred. We now have 748 suspects. You are the investigator. How do you determine who is guilty? And you have access to, say, Scotland Yard, all these, criminal databases. What's the first thing you would do? Who would you think would be guilty out of those 748?
Prior offenders.
What's that?
Prior offenders.
Prior offenders, criminal history, right? This is where you get to that. Go to disease and function. This is my idea of psychedelic. Here's what I want you to do is go by number of genes. This is a criminal history of cancer. And I'll tell you right now, I've looked at pituitary tumors, I've looked at lung tumors, bladder tumors. They pretty much kind of look like this as far as criminal history. The bigger the box, the more genes from our gene list are contained in that. Not only that, but this software is very smart. Like I said, it's probably the deepest, well-curated database in the world. So what it does is it says out of those 265 genes that are associated with cancer, based on the expression or the direction of those genes, it'll make a call on whether that function is more active or inhibited. The more confidence it has that thing is activated, it'll be more orange. The more confidence the software that it has that it's inhibited, it'll be blue. And we can click on any of these things and get those genes from there and see the papers where it says that. What's the only thing going down pretty much? Cell death, right? Does that make sense? Does all this make sense? Totally, right? Growing, blood vessels, cellular development, your immune system is up. This makes sense. And this is what I typically see. So I don't spend usually a lot of time on this page. I mean, you can. It's kind of cool. We can look at quantity of cells. I thought this was really cool. They had, it's not in there. Never mind. Okay, so this is our criminal history. This is whether we got a bunch of criminals in our gene list. And sure enough, yeah, there's some criminals here. Totally. What else would you want to know? So now we know there's a lot of criminals. What about those criminals would you like to know? You're an investigator. Bunch of criminals in front of you.
What have they done in the past?
What have they done in the past? What if they're working together? What if they're in gangs, right? And that's exactly what we do with these pathways. These are our gangs. Click on canonical pathways. These are the gangs that we know of that are operating in our system. A gang associated with T helper cell differentiation. Antigen presentation pathway. If the line is above this bar here, this is the statistical threshold. Any line above this means I have more genes than I would expect by random chance. Let's go down and see if our STAT3 pathway is here. So if you just kind of go down, it'll be like, I think it's the first orange one. It's right under axonal guidance signaling. Everybody see that? It's kind of way down there. Let's click on that. Double click on that line. All of the genes that are colored in were on our gene list. If it's clear, it was not. Basically, what this is doing, you can zoom in. We can take these things. We can move them anywhere we want. This is how I get my high school kids to do this stuff. I'm going like, it's Minecraft except with genes. And they go, oh, cool. So they start moving stuff around. But you can move this any way you want. So basically, BCL2, some PIM1. If there's a double line, that means it's a receptor. And you can actually go in here and click on that, right-click. If you select it, right-click and go Show Members. It'll show all the members. And anything that was colored was on our gene list. And this is where I might be looking. Okay, we know STAT3 is important in this cancer. Could one of these growth factors be inducing it? Growth receptors be key. But you can see, and I'm going to tell you, I'm going to be honest, I don't like pathways. I think most of these things were developed like 10, 20 years ago in like a bunch of different organisms and diseases, and I don't think they're very relevant specifically to your system. These are the idealized pathways. It's just like police work. The better the police work, the better idea of the gang. Some of these gangs have been neglected for many, many years. And a lot of the gangs don't do what they say they're supposed to do. But here's what I'm saying is, this is just kind of this generic pipeline thing. What I'm going to do is I'm going to tell you how to find your own genes or make your own pathways. And I think this is what IP allows you to do, and that this is the thing that makes it very, very special, is I can create my own pathways around whatever I want. We're going to do that now. All right? So go to upstream analysis. Okay, staying with a criminal analogy, right? You've got people with criminal history, they're in gangs, but don't these gangs usually have bosses, right? Godfathers, yeah? Do you think the godfather is around the crime when it's being committed? Oh, heck no. Hey, I was at the movies. I don't know what you're talking about. That was a terrible Brando impersonation. I apologize for that. I'm sorry. No, they're not. And actually, that's exactly how it works in your cells. I'm going to tell you is that the most important things in this system that we are measuring, we don't measure directly. That the critical functions in your cell are driven by things that change post-transcriptionally. That P53 is in a huge molecule in just about every cancer, but I never measure it directly, ever, unless you drop a nuke on it. What happens with P53 is you change its activity. You phosphorylate it. You remove a nucleus or an inhibitor. You take it to the nucleus. TGF-beta, VEGF, the same thing. TNF, I never see them change at the transcript level. But based on this software knows what it interacts with, and it'll find, based on what it's, oh no, don't do that. Based on its known targets, and those targets being overrepresented in our gene list, it'll find me those godfathers. I'm going to show you how that works. It's really, really cool. All right.